Hello, welcome to Converging Dialogues. This is Xavier Bonilla. On this episode, I'm very happy to be talking with William Harris. Bill is a professor emeritus of anatomy at the University of Cambridge. He's the co-author of the book Development of the Nervous System and Genetic Neurobiology. He's the co-editor of Retinal Development, and he's also a fellow of the Royal Society. He is the author of the latest book, Zero to Birth: How the Human Brain is Built. And that's what we talk about in this conversation. We start by talking about an overview of the evolutionary history of the brain, the rise of the neuron, genesis of the neurotube and the neuroepithelium. Talk about neurostem cells to neurons, 4 cycles of the cell, neuron theory, growth cone and stepping stone cells, cell death, neuroplasticity, and the uniqueness of the human brain. I have to say, this is not a topic that I talk about often, and I wish I did. The brain has always fascinated me. Obviously, Bill is wonderful on this topic and really shows the importance of understanding how the human brain is built and develops in utero. I had so much fun talking about this stuff. A past interest of mine was in neuropsych and all things the brain, and so my mind just kind of clicks over in that way. And so I was very, very happy to talk to him. And his book is very, very good and very, very accessible for people to get a really good understanding of the beginnings of the brain and formation. And so now I bring you William Harris. I'm here with William Harris. Bill, thanks so much for coming on the podcast. I'm very excited to talk to you about your fabulous book. The book is Zero to Birth: How the Human Brain is Built, and it's quite marvelous. It's really, really nice, and so I'm excited to talk to you about all this.
Great. Well, it's good to be here, and I'm happy to talk to you too. Thanks for doing all this podcast stuff. It's really interesting.
Yeah. Well, I think it's great because you get I kind of say that it's kind of my lazy way of not writing probably as much as I should. I do, but not as much as I should. And so I just love having conversations with people that are doing really good research and good science and trying to communicate that. And you definitely have done that. So before we just jump right into it, just briefly give us your kind of overview of who you are and what your research and expertise is in, and what you're, where you're at, what you're studying now.
Yeah. Well, I'm a developmental biologist at Cambridge University, professor there, and I've been in the field of developmental neurobiology for about 30 years, and I've been privileged to witness great progress in my time as a developmental neurobiologist. Now, I've written a textbook about the subject, which is taught at universities for undergraduates, second and third year undergraduates. But the aim of this book was to provide a relatively up-to-date science-based understanding of the successive steps used in the construction of a human brain that is accessible for non-experts, non-biologists, a wide audience. And, it's because I talked to hundreds of scientists and hundreds of lay people over the years, and it's really exciting to try to talk to people about the big discoveries that are being made in this field. And most people don't know much about how brain development happens, so it's exciting.
Yes, it is very exciting. I feel like many people get the kind of, what's the popular book? What to expect when you're expecting. That's a very popular book when women are pregnant, and you kind of just go through the certain weeks, of development in utero and stuff. And so, but not enough. Even for myself, when I took biological basis of behavior and other courses, we would talk about the brain's development, the neural tube and stuff, but it wasn't really as deep as maybe it would be to understand how really the brain gets to where it's at. And of course, that's important because one of the things about human brains is that it kind of separates us or makes us different in some ways from other primates and from other mammals and from other organisms. So I think it's important trying to understand the development.
Yeah, certainly we don't look exactly like any other mammals. We look human. So every part of our body is human. Every cell in our body is human. And our brains are particularly human too.
Mm-hmm. Yeah. So let's just give what's the kind of overview, the arc of the story of the human brain, kind of from a developmental and then also tied in with that an evolutionary framework.
Yeah, that's a good question. Well, I say that, written in our genes is the whole history of human brain evolution. And it's used, that information in our genes is used to construct a new brain in every baby. And the fertilized egg of an embryo is like is like evolutionary ancestors of the first cells on the planet. They had a cell membrane, and inside the membrane was a nucleus, and inside that nucleus were genes and the genetic material. And many of the instructions that are written in the human genome were actually written 4 billion years ago when the first cells started. For example, the excitability of a membrane, which neurons have, the actin and myosin, which control the shape of cells, the receptors on a cell, to chemicals, to light, to touch, they were all invented 4 billion years ago, probably. And we still keep them. And that's part of what makes a neuron a neuron. But multicellularity in terms of brains and real neurons couldn't have arisen until about 700 or 600 million years ago, when animals first became multicellular. Before that, there was a huge eons of protozoan life. But as soon as multicellular animals came about, cells could specialize. Some cells could be for movement, some cells could be for processing information, some cells could be for digestion. Before that, single cells had to do it all within a single cell. But as soon as you got multicellularity, then you got this ability to specialize. And that's when neurons and brains started to arise. And then if you continue to trace that and you say, well, who are our ancestors? Well, our ancestors were earlier mammals. And earlier mammals had brains that are kind of like us, that we can talk about that later, but they're composed of the same kind of pattern: forebrain, midbrain, hindbrain, spinal cord. And, you can go that back further to reptiles from where mammals evolved, and keep going back. And, finally you find the first parts of the nervous system way back 4 billion years ago in these early animals. But the book, through the various chapters, traces these evolutionary strands of brain development that parallel the ontogenetic development, the development that happens within an organism. So first it's a single cell, then it becomes a multi-cell, then things specialize. That happens in an animal as well as in evolution. So it's interesting like that. Yeah,
there's this continuity, and then there's this it's just like this eternality of sorts, right? Where you have all these parts that are just continuing on and on and on and on through, here we are 4 billion years later, which it's a cellular level, which is remarkable. It's very remarkable. Maybe.
Yeah, and if you think of the germ cells, particularly, the germ cells are the ones that go on to the next generation and the next generation.
Yeah. Maybe give us, before we get right into like the neural tube and everything, give us just kind of the overview. You kind of alluded to it, of kind of the neuron, the neural stem cells, glia, what is the development of the neuron in the embryo?
Okay, well, first you have an embryo, say a fertilized egg. And that fertilized egg is going to give rise to every part of the body, including the brain. Those cells in the early embryo, up until at least the probably 32 or 64 cell stage of the embryo, are pluripotent. They can give rise to all the tissues. But at some point in development, some cells start to specialize. That doesn't mean that they're neurons yet, but they've become committed to give rise to the brain, as some other cells have become committed to give rise to muscle, and other cells have become committed to give rise to skin. And we call those the neural stem cells, the muscle stem cells, the epidermal stem cells. Those cells are still in the process of dividing like crazy, but eventually they will give rise to particular tissues and organs. So first there's an embryo with pluripotent cells, then something happens, and neural stem cells arise, and they give rise to the brain. The neural stem cells divide and divide till you get enough cells, basically, and then they start producing neurons. And as they produce neurons, they divide. Maybe one of their daughters is a neuron, or maybe both of their daughters are neurons. And as stem cells divide and produce neurons, they become more limited as stem cells. When a stem cell divides to produce two neurons, neural stem cell divides to produce two neurons, it no longer exists, and just two neurons exist. So eventually the neural stem cells give rise to all the neurons, all the 100 million neurons or so that we generate in our brain.
Now, this is still at the fertilized egg bit, and as it goes through, I guess in terms of development, but there's a space where in terms of the neurons that go for the neuron stem cells that go to be in the brain, right? So there's the formation of a brain for these neurons to essentially live in or to reside in. So I guess one of the big things is the neural tube, right? Which is where we see that development. So in the book, you lay out, or you talk about Mangold and Speeman's 1924 experiment with the two neural tubes. So maybe just kind of tell us about that and how that helps us understand what the neural tube is, how it expands, and then how do we eventually, we'll get to a brain.
Yeah. So Speeman was interested in finding out when it happened that embryonic stem cells that could give rise to everything became specialized. And so he started to transplant little groups of cells from one embryo to another. And at early stages, when cells were pluripotent, no matter what he did, when he transplanted one bit of tissue, one group of cells to another embryo at a different place, nothing special happened. The embryo just looked like it developed normally. Like the cells that were transplanted into a different place assumed a new fate based on where they were. Until one day, he did an experiment at a slightly later stage of development called the gastrulous stage. And when he transplanted this little piece of cell, he found the first cells that seemed to have made a commitment to do something, even though they were transplanted to a different place. So they were committed. But it was more amazing than he ever thought, because what happened was that a whole new embryo developed around this few transplanted cells. And, he might have expected the few transplanted cells just to be a little bit of ectopic tissue that was different, but it seemed to organize the donor tissue that was near the transplant to form a whole new embryo, including a whole new nervous system.
And so you talk about how there were two neural tubes, yeah?
Yeah, that's because there was the normal host one.
And then it branches off.
And then the transplant made a second one.
I see.
So the transplant, it didn't really make it itself. It organized tissue that was around it. So that was really a fundamental discovery in all of developmental biology, really, that one tissue could organize cells around it to do such a complex thing. And one of the things that transplanted bit of tissue did was it released a factor that caused pluripotent stem cells to become neural stem cells. And it'll probably release other things that organize other bits of the developing embryo.
How does this help us, I guess, understand the, I'm going to say this wrong, the neural epithelium, and why does that come in week 3? So as you've been talking, I've been wondering to myself, how do these things know to do these things, and how do they know to do them at certain stages? Not too early, not too late. In terms of evolution, how does it know? Maybe is not the right term, but how does it know to do this and all that? So what is the neural epithelium, and why in week 3 does that arise?
Okay, the neural epithelium is basically a part of the embryo that's become the neural stem cells. So due to this action of this tissue, which Feynman called the organizer, and it's really the dorsal mesoderm, if that means anything to anybody, but due to the action of this neural inducer that's released by this dorsal mesoderm, the cells that were pluripotent now become neural stem cells, and they're all packed together in an epithelium, a sheet. Mm-hmm. And that is the neural epithelium. It's basically just neural stem cells that are dividing like crazy. Starts out as a flat sheet, but it rolls into a tube by lifting up the edges at the side. And when you have a neural tube, it's just full of neural stem cells that are dividing like crazy. Mm-hmm. And that happens in a mammal. Well, it happens in a human at about week 3, that this neural induction process happens. In frogs, it happens, in day 1, basically.
Sure, yeah.
But it happens in different rates, different animals.
So the very early kind of, I guess, it really is kind of the neural tube that we understand that arises. It's super early. It's the 3rd week of an embryo. Is there any? I mean, this might be an unknown answer here, but why so early? Why would it in humans? Why wouldn't it be week 6? Or is there just too many other things that are developing that this needs to happen so this can happen? Or we just don't know why it's why not week 2 or 2.5? Or why is it kind of in this time frame if we know?
It's really hard to answer these why questions.
Yes.
But it does take time for development to happen. Mm-hmm. First, one thing has to happen, and then the next thing has to happen. In a human embryo, it's a little bit more complicated than a frog embryo because you have to make placental structures and extra embryonic structures. Mm. That has to get kind of established. Mm. Then the embryo itself starts to do these things like gastrulate, which happens at about week 3 in humans. But as soon as gastrulation happens, where the mesoderm talks to the overlying tissue and tells it to become neural stem cells, that's when you get the beginning of the neuroepithelium.
Yeah, no, it's super helpful. It's always interesting to me how these things have like a kind of system, and it just goes, and it's doing all these things one right after the other. Yeah, it's very incredible. You talk about the phylotypic stage. Am I saying this right? And so in there is
phylotypic, I would say.
Phylotypic, yeah, and you talk about cephalization and Hox genes. So just tell us what this stage is and what's going on there.
Okay, so people say that there's a stage of development in a group of animals that looks where the animals look very similar to each other. And this isn't the adult stage because it's easy to tell a monkey from a bird, from a human, and a reptile at an adult stage. Mm-hmm. But if you look at the tail bud stage, just soon after the neural tube forms, it's hard even for an expert to tell which is which because they all look so similar. Mm-hmm. If you go back very early in development to the egg of a bird looks different than the egg of a mammal. So things can look different at the very beginning, and they can look different at the adult stage, but they're somewhere in the middle, which is probably at the tail bud stage for vertebrates, where things look most the same. And the nervous system is the thing that looks, the neural tube looks most the same in all these different vertebrates. We call that the phylotypic stage because even though vertebrates are a subphylum properly, the neural tube, the phylotypic stage is the stage where things look most similar. And the neural tube is probably the most phylotypic of any of the organs that are evolving, or that are developing at that time.
Is this just the power of evolution, right? That there's enough pieces across different organisms that have a similar or can have similar uses of function at one stage, that it could be, obviously it's going to change maybe as it goes, as the organism is evolving, but is there somewhere where evolution can help us explain why there's such similarities in this stage, or is it just, kind of by happenstance?
Well, I think even Aristotle recognized that embryonic animals looked more similar than adult animals. And then there was a very famous biologist named von Baer who put together some laws of embryology. And his first law was that at earlier, common traits develop first. So the thing we have in common with all vertebrates is a brain with a basic similar pattern: forebrain, midbrain, hindbrain, spinal cord. And you can see that developing at an early time in all vertebrates, and it looks pretty much the same.
In terms of the regions of the brain, right? So after we have this stage, we have many different locations, right? You have head to tail, back to belly, inside, outside, left, or all these different orientations. So how did the different areas of the brain start to rise, I guess, outside of week, 3 going into, the different, weeks, week 4, et cetera? How do we start to see these different locations start to begin to form?
Yeah, so probably at week 4, when there is a definitive neural tube, it is already patterned, even though the human embryo is about the size of a sesame seed at this time. If you look under a microscope and you look at the little neural tube that it has, you can already see a bulge of forebrain, a bulge of midbrain, a bulge of hindbrain, and a spinal cord forming. And the neural tube has a top side and a bottom side, or a back to belly side. And that is also known as dorsal to ventral. It also has, a head to tail thing. So you've got the forebrain, midbrain, hindbrain going from the head to tail region. So those first boundaries between different regions are set up. And they're set up on 2 axes, kind of Cartesian, X coordinate, Y coordinate. And you can consider the neural tube as a kind of a checkerboard of areas. And in this checkerboard of top to bottom and front to back structures, you've got little regions that express different genes, particularly, as you mentioned, the Hox genes. They come on in particular little domains, and they're regulated by gradients of molecules called morphogens that are percolating through the embryo at this time. So maybe some molecule is very high at one end of the embryo and low at the other, like retinoic acid. Retinoic acid is high at the tail end and low at the front end. And where it's high, it turns on some of these Hox genes, which are transcription factors, which turn on tons of other genes, which say be part of the spinal cord and be near the tail. And at low levels of retinoic acid, which are near the front, it says don't be spinal cord, turn on Hox genes that are part of the brain. And, then there are other ones that say forebrain, midbrain, hindbrain. And these genes come on in because they're at particular levels of these percolating morphogens that are running through the tissue and telling the cells about their positional information inside this tissue. So the brain is highly patterned at the neural tube stage. Even though it's tiny, it's got hundreds of little regions already. And these regions turn into things like the cerebral cortex, eye, cerebellum, so forth.
Yeah, it's really astounding how I didn't know until reading your book that, all of this groundwork was laid at week 3. I knew obviously there's,
week 3, 4, yeah, somewhere around there.
It's really incredible.
But it keeps on going because the brain gets bigger and bigger.
Sure.
And as it gets bigger and bigger, new regions are formed. So there was a big region before, like a big country, and then it split in 2, and now it's 2 countries. And that's because new morphogens have come on and turned on new genes that separate the front part from the back part.
Also at week 4, I think you mentioned this in the book, there's, how do we understand this kind of neural proliferation? Where's the role of glia cells and myelination? Because obviously these are really important in this kind of expansion for these different regions. How are these pieces implicated?
Okay, so we've got a neuroepithelium, right? It's a neural tube. It's in the shape of a neural tube. We don't have any neurons yet. All we've got are these neural stem cells, and they're dividing like crazy. And one neural stem cell divides, makes 2 neural stem cells. Those 2 neural stem cells divide and make 4, and the 4 make 8. And they're just dividing and making more and more neural stem cells. By the time an embryo is about the first trimester, it's making, I think, 15 million cells a second or a minute or something like that. An astounding number of cells. At some point, you have to make neurons. You can't just make neural stem cells forever and grow forever. The brain is a particular size. So the neural stem cells slow down and they divide in a different way. Now, sometimes they divide and one daughter stays a neural stem cell and the other daughter leaves the division cycle forever and becomes a neuron. And then near the end of development, which is near when birth happens, the last neural stem cells divide, and they divide to make 2 neurons. And then you don't make any more neurons after that. Basically, your brain has got all the neurons it can ever have. The neural stem cells, those that are left, they're no longer neural stem cells. They become glial stem cells, and they make some glia. And glial genesis, the birth of new glial cells, continues postnatally. But neurogenesis, the making of neurons, ends pretty much at the end of, at the time of birth.
And how do these divided neurostem cells eventually contribute to functioning in the cerebral cortex? I mean, this goes all the way up to the cortex. How does this? How are they contributing? So they're dividing and then they're making neurons. Is there any other role that they have?
The neural stem cells. No, that's their job is to make.
They're done.
The brain. Their job is to make the brain. They don't seem to have a survival function in the embryo itself. So their job is to make the brain. If they don't make a brain, of course, the embryo is not going to survive.
Right.
So they make the brain, and the cells that are in the region of the neural tube that make the cerebral cortex, the neural stem cells there, make cortical neurons. And the neural stem cells that are in the region of the retina make, the cells of your retina, the rods and cones and other cells of your retina. The neural stem cells in your spinal cord make motor neurons and other cells that, like the motor neurons, are used to activate the muscles. Right. So different stem cells in different regions of the brain make different types of neurons.
So they're definitely the precursor that kind of get it started. And then once they get to neurons, their job is done and they're no more.
Yeah, they're gone, basically.
Yeah. And how does, I guess, neurons they're born in the first weeks of gestation. Is this correct?
The first neurons are born.
Okay.
But neurons are born throughout. From about, I'd say the first neurons are born probably at 5 or 6 weeks. Those are the very first neurons. probably in the hindbrain. But neurogenesis continues all the way up until birth. It becomes more and more. So during the second trimester and the third trimester, more and more neurons come out of the neural stem cells. So at first it's just a trickle, but as development goes, as embryogenesis goes on in utero and mammals, more and more neurons get born. But by the time of birth, it slows down. It is slowed down to basically a halt, except maybe for one region of the brain, which is the hippocampus, part of the hippocampus.
Yeah, it's interesting how there's like it's kind of a slow hum, and then it just has this explosion, and then and then and then you get to birth, and then there you are. It's fascinating how the brain is doing all this in development. How the brain is developing is starts to become quite quick, is what it seems like in the second and third trimester.
So I think it's really important for people to think about the fact that when a baby is born, it has all the neurons it will ever have for the rest of its life. More, in fact, because some of those die. A lot of them die, actually. But most of the cells in the brain, 99.9% of the cells in the brain, the neurons in the brain, have already been born by birth and have started to connect up.
Maybe kind of backtrack a little bit here before we push forward. I didn't ask you about the 4 cycles of the cell cycle. I don't know if this is important to kind of go over for this bit, but what are the 4 cycles of that cell cycle, and where the checkpoints fit in, and all the asymmetric and symmetric cell divisions? Maybe just tell us about that a little bit.
Cool. Yeah. So in all organisms that I know of, we can divide the cell cycle into 4 stages. And that's kind of a scientific usefulness, but it's a continuum, really. Basically, a cell grows, a dividing cell, it grows, and then it divides into 2, and then it grows again and divides into 2. So in the first phase, it's a growth phase. Basically, the cell is sucking up nutrients, importing things, getting bigger, getting the materials that are enough to say, okay, I've got enough material that I can divide into 2. I'm big enough. I'm ready. And then what it does with those materials, it does in the second stage of the cell cycle, which is called the S phase, or the synthesis phase. It replicates all the genetic material and all the other necessary stuff that it needs to separate into the 2 daughter cells. And in the third stage, it starts to line up the materials and grow a little bit more and check that everything is being replicated properly. And then the fourth stage is the final stage, called mitosis, which people have heard of, I suspect, which is when a cell divides into 2. And half of the genetic material goes to one side, half goes to the other side. So it's being replicated in S phase, and now an equal amount goes to each side, 1 copy of every chromosome. So growth phase 1, synthesis phase, when you replicate things, growth phase 2, when you check that things have been replicated properly, and then dividing into 2 cells. And there are these checkpoints between these phases. So between the growth phase and the synthesis phase, the cell says, have we really got enough material to go through the whole thing? Or should we just abort and just stop the cell cycle because we're not big enough? We don't have enough stuff to do it. So that's a checkpoint. And there are little molecules and proteins that ask that question and answer that question, allow the cell to go through the next stage. And then once it's replicated stuff, you ask, have I replicated the DNA properly? Are there any errors? So you check that. And if that's okay, you go to the next stage. So those are the checkpoints.
It's interesting how, again, there's a system of how every cell is doing this, right? Which is incredible to me. It's just how we understand at this point in life, billions of years later, how we were able to do this with our cells are able to do this, and they do this with big organisms, and it's always incredible.
Well, even the protists at the way back 4 billion years ago had most of these components of the cell cycle. So that's ancient. Mm-hmm. And what's interesting about the nervous system is that, you have to make a brain of the right size. Mm-hmm. So you have to know, in normal development, how big do you want your cerebral cortex? How big do you want your eyes? Right. These things. So they're regulated in a region by region basis. Mm. And the same thing that patterns this neural tube, mm-hmm. the same molecules and genes that pattern the neural tube also kind of specify how big they want that structure or region of the brain to be.
Mm. Yeah.
Regulate when a neural stem cell goes from symmetric proliferative division to an asymmetric division where it makes one neuron and one neural stem cell, or to a different kind of symmetric division where it makes two neurons. Mm.
Yeah, well, I can imagine that here the proportions become really important, right? In terms of like, how big do you want your cerebral cortex? How big do you want your eyes? Like, these things have to have the kind of the script of sorts of saying like, okay, here's the plan, and let's go and do it. And so it's interesting how that's happening at such an early stage in development. I guess
it happens at an early stage and it continues.
Yes.
Like, for example, a human has a huge cerebral cortex. So our cerebral cortex, what do they say, is like 3/4 of our brain, mm-hmm. the mass of our brain is cerebral cortex. It's dominant. In a monkey, it's more like half of the brain is the cerebral cortex. Mm-hmm. In a mouse, it's more like 1/4 of the brain is the cerebral cortex. Mm-hmm. Bigger cerebral cortex is part of being a mammal, it seems, because that's what seems to have expanded in mammalian brains, but particularly human brains. Mm. So these neural stem cells, if you take a neural stem cell from a mouse and you put it in the same conditions as a neural stem cell from a human, then it will make a smaller batch of cells. Mm. And a human will make a bigger batch of cells. Mm. So somehow these neural stem cells are programmed to know how many times to divide and how many neurons to make. And that's an evolutionary mystery.
Yeah. Well, this would be a good point here to kind of tell us about the Neuron Doctrine, right? Can you maybe tell us about the Neuron Doctrine, and then maybe after that you can tell us about Ramon y Cajal, because it's super important.
Well, the Neuron Doctrine is what most neuroscientists now believe, maybe all neuroscientists now believe, which is that every neuron in the brain is a single cell. Before the Neuron Doctrine, it was assumed that maybe the nervous system, because they already knew it was kind of electrical. Galvani had done these experiments where he'd, use a lightning bolt to excite a nerve, a sciatic nerve of a frog, and saw the nerve twitch. So he knew that electricity was running through the nervous system. Mm. And that's what made it work. But the old histologists, they started to, how does this nervous system work? And they started to pull apart the cells, and they saw that each cell had this fine wire coming out of it, the axon, and has other wires like the dendrites coming out of it. Mm-hmm. And they tried to dissect these, and they could never find the end of it. Mm-hmm. So, and they knew that electricity was flowing, so they thought, well, electricity has to flow through wires, so there can't be a stop from one cell to the next cell. How would the electricity get through? Mm-hmm. So that was the sensitial hypothesis for how the nervous system worked. And it had a lot of credit in the early 1800s, particularly by a guy named Camillo Golgi. And then came along Cajal, this guy, Ramon y Cajal, and also an English physiologist named Sherrington. And Sherrington noted that, when you have a reflex action like pulling your hand away from a hot thing, some muscles contract and other muscles relax. How could that be? How could it be that some muscles contract and other muscles relax, unless the sensory signal that come into the neural tube didn't excite everything the same way? It excited the motor neurons that went to the muscles that contract, and it inhibited the motor neurons to the muscles that relax. So he thought, well, there must be some way of getting a plus minus signal into the system. And he thought, well, neurons could be doing this because some neurons could be exciting other neurons, and other neurons could be inhibiting those neurons. So he proposed the idea that there were synapses where cells talk to each other. But he had no physical evidence. But Ramon y Cajal was a histologist, and he used techniques that were developed by Golgi, particularly silver staining techniques that would fill a neuron to its entire edges. And he saw that neurons of a certain type all looked the same, and they all had endings. And the endings looked a little bit like what Sherrington would have thought were synapses. And there was no continuation. There was no, just things flowing into each other. Neurons had real boundaries. But it wasn't until the invention of electron microscopy that was actually proved, that it was found that every neuron was completely surrounded by a cell membrane and was an individual. So then the other question you asked me is about Cajal and the individuality of neurons. Mm-hmm. Well, once Cajal had figured out how to stain neurons in their individuality, in their wholeness, basically, and he could see the whole thing, except he still couldn't see a whole neuron and its axon if its axon went very far away, because he was just looking at slides. But he could see that they had an axon that was going somewhere. But he could see that the shapes of neurons were very different. Motor neurons look one way, rods and cones in our eyes look one way, ganglion cells look another way, cells in our cortex. He maybe described 50 different kinds of cell shapes in our cerebral cortex. And he described hundreds of different types of neurons. The nervous system, the brain, has more different cell types than any other part of our body. Maybe some would argue the immune system has that, because you have these cells that produce this antibody or that antibody. And maybe those are different types of cells. But, they look the same. But different neurons process different information in different way due to the way they're shaped. And people don't know how many types of neurons there are. There may be thousands, there may be tens of thousands, there may be millions or billions. In fact, there's some good argument to say that every neuron is an individual, knows who it is, can distinguish itself from others. And, it's just amazing.
Yeah. Well, it's mind-blowing. And it's also fascinating to hear, Ramon y Cajal, the tremendous significance of making this kind of discovery, which is what we still understand today about neurons. They go down the axon, they get to the, presynaptic, and then they have the synapse and postsynaptic. It's very, that stuff is still super helpful for us today.
Sure. Ramon y Cajal he wanted to be an artist. And he ended up one should read his biography, his autobiography called Memoir, I don't know, Memoirs of My Life or something like that. Mm-hmm. And it's a fascinating book, and he describes his life. But he wanted to be an artist, and he was a very good artist. And what he did was he actually, saw these neurons in his microscope slides, and he drew them beautifully. And he wrote this book called The Histology of the Nervous System of Man and Vertebrates, which is still used today by neuroscientists around the world because they're the best pictures of different types of neurons that are still yet available.
Oh, yeah. I mean, that's one of those things where after, you're gone from the planet and people are still using your stuff and still talking about you and still using your contributions to science. It's super awesome.
And he and Golgi both won the Nobel Prize together.
Sure. Yeah.
But they hated each other because Golgi still believed in this syncytial theory, even though the rest of the world had moved on. And, they were bitter rivals as they received the Nobel Prize.
That's very funny.
Yeah.
Into the second part of your book, you talk about kind of the, basically how do neurons connect? How do they, wire up, and how do they run, and how do they do all these things? And so I guess the first question here is how do neurons know how to wire up? I mean, again, this is maybe a sort of similar to the why question of sorts, but how can they possibly know how to connect and to wire up? How is that possible?
Yeah, that's a great question. Certainly, it's true that a young neuron has a sense of who it wants to make connections with. It has already an identity. It knows it was born in the cerebral cortex, for example, or the retina.
Yeah.
And it knows it was born with a particular fate in mind. Perhaps it's a motor neuron in the spinal cord, and it wants to innervate a specific muscle. I say wants to as if it was a human, but it's of course not. But what it has done is it's expressed certain factors, certain genes, maybe transcription factors and other genes that help it know who it is and how it should grow. So it may be sensitive to a particular guidance factor that's out there in the environment and say, okay, I'm interested in that. I'm going to grow towards that. And it does that. And it finally grows to the right place because it navigates, it has a sense of where it wants to go. And the growing tip of the axon, the growth cone, which we'll probably talk about in a minute, is reading the cues and growing along to a particular target. But it knows that from the beginning that it wants to go there.
Well, I think that's the thing that is like so interesting about that is that many people, you can kind of see how people have this idea, these people that are, do panpsychism, right? Where they think that there's sentience in every cell in the body and it has its own like kind of, which, some people kind of, brush that aside and say that it's kind of woo-woo kind of science and things like that, which it certainly sounds pretty spectacular and maybe not the best ways, but it is fascinating how there is this kind of like, hey, this neuron is the individuality of the neuron and saying, hey, I'm in this region, I have this function, or I'm going to do this thing. It's really surprising to me to kind of hear in some ways that it's that individualistic.
Yeah, it is. It's amazing to me too. I mean, one of the things that was discovered fairly recently is that there's these molecules that are on the surface of neurons, cell adhesion molecules, and certain genes can be rearranged in particular ways, or the messenger RNA of those genes can be arranged in certain ways so that a particular cell may express one of several million versions of this gene that are randomly assorted, just randomly stitched together. This cell, have this lottery ticket. This cell, have this, or you could say barcode. And they each get a different barcode, even though they might be the same basic type of neuron, but they have different barcodes. So they can tell themselves from others of the same type. And that prevents them from making like synapses with themselves because they can recognize themselves.
Yeah. Again,
So every neuron is different, and it's also every neuron in your body is in your brain is at a different place. Mm-hmm. So in your retina, for example, some are at the top of your eye, some are at the bottom of your eye, and those don't want to make connections to the same place in your brain necessarily if you want to keep a map of space in your brain. Mm. So even though they're the same type of neuron, in a way they have a different positional identity from each other. Hmm.
Yeah, it's incredible, and especially kind of what you were saying, recent discoveries. I mean, that's the thing that's so fascinating is that, what is it? Neuroscience is changing like every 6 months or 18 months or whatever. It's just new stuff we're finding out, which is incredible because then you kind of have to maybe not entirely redraw everything, but you definitely have to consider like, oh, we have to add this feature now or this variable to what we already know. Does it work? Does it fit? Does it not? And that's exciting and interesting at the same time.
That's true, but it's also too much hype for me because I think, the book I wrote was about what I think are the most basic, mm-hmm. well understood principles and processes involved that have been tested. And this book isn't out there about their very fringes of things very much.
Sure.
Well, I wanted to, get the basics. What isn't changing? What is mm-hmm. pretty established out there? Because most people don't know that either.
Mm-hmm. Well, certainly, especially when going back to the idea of what was, around, or what's in development at 3 and 4 weeks. It's incredible how we, that has happened for, years and years and years and years, obviously. And so it's like, okay, we understand what happens at 3 and 4 weeks, which is helpful because then you can learn how to, when you're trying to do kind of proactive kinds of care and what's important. And okay, this is what's going on here at this stage of life or in development is super, super important.
Yeah, particularly, for example, mini brains. I talked about mini brains in the book, mm-hmm. which are a fairly recent thing. But over the last 2 decades, people have learned that you can reprogram almost any cell in your body to become a pluripotent stem cell again. Mm. All you have to do is, activate the right genes, and it will start to reprogram itself. And when it's a pluripotent stem cell, you can grow it in a petri dish and make tons of them. And then you can induce those cells to become neural and turn them into a little bit of neural tissue. And then with these morphogens, you can say, okay, I want it to be anterior dorsal neural tissue, like the cerebral cortex, or I want it to be posterior ventral neural tissue, like motor neurons. And you can, in a tissue culture dish, by the things we've learned about developmental biology, make cells turn into different types of neurons, different parts of the nervous system. If you can do that from patients with, say, a motor neuron disease, maybe you could find out what's, more about what's going wrong there. Yeah. Or maybe you could grow new motor neurons to replace old ones, or new dopaminergic cells to replace dying ones. Yeah. So there's a lot of usefulness, mm-hmm. you could say, in understanding the developmental program in order to advance medicine as well.
Yeah, certainly. Yeah. And I think that's, again, absolutely essential. We mentioned the growth cone. Tell us what the growth cone is and the importance of actin filaments.
Yeah, so the growth cone, when a neuron is born, it's just basically a round ball, and it isn't connected up yet. It's never going to divide again, but it still hasn't turned into this neuron that saw, with its elaborate morphology. So it has to grow all these fine processes. Some are dendrites, which receive information. And one process, usually it's only one, is an axon that grows a long distance away from the cell, usually, and connects to other neurons in a different place in the brain. Mm-hmm. So when an axon starts growing, when a cell starts putting out an axon, It's just a process that's ticked with something like a structure like the hand at the end of your arm. And you could think of the hand as a kind of a spread out structure, and the fingers of your hands as little antennae that are feeling in different directions. And inside those little antennae are also little cables of actin, which are molecules that form cables. And there's myosin, same thing that's in your muscle. So actin and myosin make your muscles contract, and they also can generate force in a growth cone. So if a Phylopodia touches something, if one of these fingers of your hands touches something or smells something or senses something that it wants to grow towards, it can activate that cable and pull it in, haul in the growth cone in that direction.
And how does the stepping stones cells, how do those function in here as well, or how do they function here?
Yeah, stepping stone cells are just a kind of an illustration. So this is all in chapter 5 of the book about how axons grow and navigate, and they make some remarkable journeys. And I guess this is a good point to just say that, the whole story is told kind of from the perspective of neurons. First, a neuron, his ancestors are neural stem cells, and they're getting their instructions about what part of the body or what part of the brain to be. And then a neuron is born, and then it has to do all these things. It has to grow an axon to a particular place, and it has to grow dendrites that connect up. So this stage of development is where a neuron is sending out an axon and growing to a particular place. I forgot what your question was.
Well, no, I was just asking about the stepping stone cells and how these function here.
So when people were interested in this, they said, well, how the hell are we going to study the way that axons grow and make specific connections? If we did this in the relatively mature brain, it's really complicated already. There are billions of cells there. There are zillions of axons that are going every which way. How are we going to? So it was actually Cajal who said, well, why not, study this at the very earliest time when the first axons are going out there in the brain? What are the very first neurons to make axons? These are the pioneers. They pioneer the pathway. And some of the pioneers were found in the limbs of grasshoppers. And these cells at the tips of the limb found their way into the central nervous system, because these are part of the peripheral nervous system of a grasshopper, by hopping from one cell to the next cell. So the growth cone liked a cell and stuck onto it. This was called a stepping stone cell. And then a little ways away, there was another stepping stone cell. And one of the Phylopodia that was on the growth cones reached out and touched this next cell. It said, oh, that's where I want to go next. So I'll step onto that cell. And it's because these cells have molecules on their surface that the pioneer growth cones liked to adhere to. So they would adhere to one, and then they would adhere to the next, and adhere to the next. And that's how they found their way into the central nervous system. And basically, it brought up this concept of cell adhesion. Later axons that developed in the limb, later sensory axons, saw that the motor neurons had already found their way into the central nervous system. And all they had to do was glom onto the pioneers by sharing cell adhesion molecules with them, and then they could follow those pioneers in. And now the brain starts to be wired up, first by pioneers that establish the first pathways, the first roads, and then other cells follow them. And they get to a place where, it's close to where their destination is, but they might have to branch off to make a new thing. But they use a highway, basically, to get close to where they want to, and then they forge a new pathway. And then later cells follow that highway, follow the new pathway for a while, and then they have to branch off too. So the brain gets more and more complicated by, first there's small roads, they grow into streets, they grow into highways, and then you have to branch off these to get all the destinations. Does that make any sense?
Yeah, no, it makes tremendous sense. As I'm listening to you, I'm reminded of how I felt when I read the book, where I just was, was feeling like, super astounded at how all this stuff functions. It's just all these things are going on in the brain, in a developing brain, not even like a fully developed brain, which it continues to, astound me.
Yeah.
Maybe.
So if we go back to just for a second to this neural tube that's highly patterned, all these regions, and you think of it as like a map of the world. Let's unfold that, put it on the table, see all the regions. Mm-hmm. Then one neuron born in a particular place wants to go from region A to region C. And another neuron that's born in region C wants to go to region D. Mm-hmm. But they're using the same map, so they're reading the same regional cues to go to different places.
Yeah. Where does Sperry's theory of neural connections fit in here? I feel this is a good place to bring him up, because he was obviously super important. Where does his theory fit in here, and why is it important for the developing brain?
Yeah, so neurons growing and navigating is basically chapter 5, and Sperry is in chapter 6, where we're talking about how neurons make very specific connections. So once they've navigated from one part of the brain to another region of the brain, they have to find the exact partners to make synapses within that other part of the brain. And for a long time, Sperry's boss, a guy named Paul Weiss at the University of Chicago, thought this happened because the neurons weren't particularly specified to find particular targets. He used motor neurons, basically. So he thought, okay, motor neurons are growing out of the spinal cord and into the limb, for example, and those motor neurons don't care which muscle they innervate. But once they innervate a muscle, say latissimus dorsi or some muscle or something like that, they get told by the muscle that you've innervated me. So now you're a motor neuron that's innervated this particular muscle. And you can tell the neurons that innervate you, I'm the guy that's innervated latissimus dorsi, so make the right kind of synapses with me. So it was thought it was kind of random first connections and then specification by who you connected to. So kind of a back propagation, if you would, of identity.
It sounds like it's the kind of relationship that's going on. The neurons are connecting with a certain part of the body, and then it's just saying, okay, we're receiving, here's what we're, talking back and forth, essentially.
Exactly. And that idea was strengthened by the experiments that were going on in Weiss's lab, where they would take nerves and cross them from, say, a flexor to an extensor and see that the limb moved in the wrong way after that. So the motor neurons could make synapses with the wrong muscle, but then after a while it got corrected and the coordination came back. So Weiss said, well, the neurons are learning who they are again because they've connected to this muscle, and we have to regularize behavior and make it coordinated. So that's how it works. Called it the resonance theory. And then Sperry came along. Sperry was a student of Weiss, and he did the same experiments in a mammal, in a rat at a very early stage, cross-innervated flexors and extensors, and normal movement never recovered. And he said, what was going on? So why do I get, is it different? Is it a difference in a mammal versus a newt, which is the kind of animal that Weiss was working on? Or is it that something else is happening in the newt? And he thought, well, maybe what's happening in the newt is the reason that normal function is recovering is because the neurons, the axons, were uncrossing themselves. Even though they were put on the wrong muscle by the experimenter, they started to grow and find the right muscle, find their normal right muscle back again. And that was actually how it happened. Now we know that motor neurons, when they're first born, already have an idea of which exact muscle that they want to innervate. And the muscle knows which motor neuron it wants to innervate it. And it's the same for the cells in the eye. So if you're born in the ventral part of the eye, you have certain molecules that say, okay, I'm going to go and innervate this part of the. So Sperry's idea was that since the retina and the part of the brain that it connects to, called the superior colliculus or the optic tectum, and that's one of the regions of the brain it connects to, it has a topography. So you have a vision of the world that's projected on your eye through the light that's coming in on your eye and stimulating cells in your retina in different places. But the axons from your retina, and they grow into the brain, and they make a map of that world on the first target structure that they enter. And they do that from the beginning, because what Sperry did was he rotated an eye 180 degrees in a newt. And then he wondered, is vision going to be upside down and backwards through that eye? And he found it was. And the animal never learned to snap in the right direction at a piece of food. It always went in the opposite direction.
Wow.
And similarly, if you cross a nerve in your face, for example, which sometimes is done in medicine, because if you have a paralysis due to nerve damage, you can regrow. But your instinctual reflex, you can learn to smile, but it isn't natural in a way. Your instinctual connectivity is still messed up in a way.
I mean, that's completely remarkable, right? I mean, I don't know how many people know this, or I mean, obviously people that are in neuroscience know this, but to the layperson, but to the layperson, I didn't know a lot of this. It's. I mean, again, there's no words almost for it. It's remarkable that the human brain and body can have this kind of interaction and all the way at the neuronal level. It's marvelous.
Yeah, there's a huge amount of specificity in the nervous system. When you think about the genome, we may have 20, 30,000 genes, maybe more if you count RNAs. The nervous system expresses more of those genes than any other organ. It's genetically really complex. Cells are specified to a high degree. They know what shape they want to make. They know what kind of physiology they want to make. They know what kind of, when I say no, I guess I'm humanizing it. It's not really they know, but
right.
They're geared to develop in certain ways so that they process certain kind of information. They connect to certain other cells.
Well, in all of these ways, we've been talking about, just the expansion, explosion of neurons, but there is something called apoptosis, right? This is cell death. And where does this factor in?
Yeah. Apoptosis, that's the way I pronounce it.
Apoptosis, excuse me. Yeah.
Apoptosis. No, some people say apoptosis, but should be a silent P if you're going with a Greek pronunciation. So apoptosis is a kind of a programmed cell death. A cell commits suicide. And it's very important in all of our organs. We wouldn't be able to open our eyes unless the cells that connected our top eyelid with our bottom eyelid died and separated the eyelids. Our fingers would be webbed unless the webbing between our fingers that developed early in development, unless those cells died and separated our fingers from each other. So cell death is happening in the immune system, in in every system in our body, and in the nervous system in particular.
Mm.
Now, some cells die because they're not needed anymore.
Mm.
They were involved in the scaffolding, building something. So you build the cortex, you start to make connections between neurons that will no longer survive once other connections are made. So they make the kind of primordial circuitry, helping other cells make the right circuitry. They're no longer needed and they die. But most of the death of neurons comes from a competitive aspect of neural development, a war between young-born neurons. You make too many neurons. You make about twice as many neurons in your brain as will survive to adulthood. Most of them die during childhood or even before childhood in the embryo. And that helps with systems matching. So let's say you have big muscles. You might want a lot of motor neurons to innervate those big muscles. So you make a lot of motor neurons. And if you have big muscles, you've got enough to innervate them. But if you have small muscles, they might not be sufficient to give nutrients, basically, to all the motor neurons that were originally born. And so some of them might die. So you always make too many of almost every kind of neuron, and they compete so that at the end, there's enough neurons of every type to innervate each other and the targets. I don't know if I've explained that very well.
Very well. Does this continue until as we get older? So after you're born, are we still having cell death or no? It's just in utero, excuse me.
Yeah. So no, in utero, there's some cell death. There's some parts of our brains that develop earlier, and they're basically finished developing and generating neurons and cell death program before we're born. But in the cerebral cortex, which is the last part of the nervous system to really develop fully, cell death goes through childhood. But at the end of childhood, basically programmed neuronal cell death or competitive neural cell death stops. And now the neurons that you have that survive this great period of competition and war with each other to make synaptic contacts, those ones that have made it, they tend to survive the rest of your life.
Wow. I want to ask about kind of the regions of the brain and come back to that. But before we do that, I want to ask about neuroplasticity. It's also something that you mentioned. I believe it's chapter 8, I think, in the book. And just tell us what it is and how does that change over the lifetime?
So neuroplasticity, what people mean by that is the ability of neurons to change their connectivity. And in adults, neuroplasticity, the most common way to think of neuroplasticity is in terms of learning. So if you learn something from reading the book and it stayed in your brain somehow, that was due to some physical change in your brain. And what that change seems to be is in the strengths of synaptic connections. So there's a ton of wiring in your brain, and you want to make a memory. And to do that, you have to change the strengths of, there's 100 trillion connections, possibly synapses. And you're strengthening some of those and weakening others. And that helps certain circuits go, maybe a circuit for this memory. So even though there's no new synapses being made, you can strengthen some synapses, weaken others, and encode something. And that is the plasticity that goes on throughout your life. You can learn new things all the time. When you're a child, a neuron has more flexibility. It's more plastic. It can make new synapses with new cells. It can get rid of synapses more easily. It can prune a whole branch away from its axonal arbor or can remove a dendrite. And so, and if you go back into even earlier times when cells are just starting to make connections with each other, that's probably when they're most plastic. So when neurons are very young, they're very plastic like humans are. You can make this friend or that friend or so on. But once you're an adult, you've made all these friends, then it's harder to change and get rid of them. You're kind of stuck with them.
Mm-hmm. It's interesting.
And then as a neuron gets even older, it goes into old age.
Right.
And maybe it's even less plastic at that point than it was when you're, a fit middle-aged adult.
Right. I was going to say that as we go through the lifespan, I can imagine that this becomes less flexible, if you will, or of sorts. It becomes
And you think about this in the terms of a single neuron. A young neuron is very flexible, like a young human, and an adult neuron is pretty much set in its ways, doesn't change that much. Mm-hmm. And then there's an old age part where things may degenerate.
Yeah. I want to move to the last chapter where it's all about the human brain and what makes the human brain special or unique or different, I guess you could say, whatever word you want to use. But I guess just before we do that, I want to backtrack a little bit. We had stopped, I guess, in terms of timeline here at like 4 or 5 weeks. Where do we get, I guess, just kind of the brief, just march us through the brief kind of place where we get to a full term is 37, 38 weeks or whatever it is, and up to 40, whatever.
40, it's called 40, yeah.
Yeah. What has developed at that point in the brain? Because there's still brain development outside of the uterus, obviously, when it, and as we're growing, right? And so we're different parts of the subcortical systems and cortical systems, et cetera, and all the way up to the common, what is it, the 25, 26 years old, your brain's fully developed, et cetera. So I guess just through utero, let's say it's a full term, what is part of the brain at that point?
Yeah, so I would say that there's a kind of a gradient of things going on. So your spinal cord kind of wires up early, and it's starting to wire up in the embryonic stages, and it's pretty much done basically, and motor neurons have connected to the right muscles and stuff like that long before you were born. But the neurons in your cerebral cortex, they're still growing and getting bigger at the time of birth. Well, all your neurons are probably getting bigger, but they're still growing and making new synapses, connecting up. All your neurons have pretty much been born, except for some cells perhaps in the dentate gyrus of the hippocampus. So they're all born at birth. Glial cells, most of them have still to be born. And they're oligodendrocytes that wrap the axons, and there's astrocytes that help make synapses and help get nutrients to the brain and do lots of other good things to help neurons do their work. So those are still being born postnatally. And your brain grows from birth to about the age of 7 or 8 till almost full size. It grows by a factor of 3. Basically, a baby's brain is small, but it's mostly the glial cells and the growth of neurons that contribute to the brain growth at that time, not the addition of new neurons.
In terms of regions of the brain, so obviously we have, frontal, temporal, parietal, occipital, cerebellum, etc.
Cortex, cerebral cortex. Yeah.
All the parts of the cortex, and then you have all the parts of the subcortical parts. So you have the basal ganglia, hippocampus, amygdala, on and so on and so forth. These parts, all of that is there, or all of this is continuing to just expand and get larger after you're born? Well,
as I tried to indicate earlier, the primordia for all these areas is already evident
in place
at the neural tube stage at about 4 weeks old. But the tissues are growing at different rates, and there's a bit of a gradient. So caudal areas, spinal cord, hindbrain develops before midbrain, before hindbrain, before forebrain.
Forebrain. Mm-hmm.
Yeah, and cortex is one of the last ones to get fully developed. Which is why I tell the story in the book from the perspective of a neuron rather than from this perspective between 20 and 25 weeks, this happens. Because between 20 and 25 weeks, this is happening in some neurons, and it's already happened in other neurons, and it's yet to happen in other neurons. So to understand brain development, it's nice to think of it from the perspective of what a neuron is going through.
Yeah, that's great. That's great. So what is it about the human brain, I guess, that makes it different or unique from our, fellow primates, our cousins, if you will, or other mammals, or just other multicellular organisms? What is it about human brains that are distinct?
Yeah. Well, humans have relatively large brains for our for being mammals. We're about 10 times larger brain, and most of that is due to the cerebral cortex, because the spinal cord and the midbrain and the hindbrain isn't that much bigger in humans in general. But the cerebral cortex is quite a bit bigger. And when you compare ourselves to chimpanzees, which are our closest relatives, our cerebral cortex is about 4 times bigger than a chimpanzee's. Now, if you look at the evolution of hominins, and the earliest guys like Australopithecus had brains that were bigger than chimpanzees, but not a huge amount bigger than chimpanzees. And then there was, I'm not an expert on human evolution, but I've read about the size of brain cases and things like that. So when you start to getting to upright man, their cortex increases again. And by the time you get to humans or Neanderthals and humans have about the same size brains. 50 million years ago, something like that. Those species, or maybe they diverged 500 million years ago. I can't remember exactly right now. But since then, the human brain hasn't grown any bigger. But basically, the human brain, or the hominid brain, and if we want to call Neanderthals, hominids, and Denisovans, those brains had cerebral cortexes which dominate the brain. So before then, maybe half of the brain in a monkey was cerebral cortex, but in a human it's like 3/4. So it's all of a sudden become the dominant structure. So the size of the cortex is one thing that's particularly human. It's not to say we have the biggest cortex. Whales have huge cortexes.
Dolphins have a very large brain.
Huge cortexes. But if you look at the cellular structure, possibly not so many neurons or the thickness of the cortex. But we really don't know enough about dolphins and whale brains to say too much about them. But compared to our closest relatives, we have thick neural cortex like cerebral cortex like a monkey, but we have more of it. So, and also the shape of our cortex is a little bit different. So Neanderthals have a slightly different brain case than we do, kind of more elongated front to back, from the front of our head to the back. So that's different in humans, in Homo sapiens. If you compare cortical areas, so people have been able to, we talk about all these regions of the brain. How many regions of the brain are there? Back in the 1800s, Rodman found like 50 areas of the brain or something like that. And now we know that there's more like 180 areas on each side of our cerebral cortex. And if you do the same analysis in a monkey, say a rhesus monkey, you only get about 160 regions. So we have new regions. We don't know what these regions do, but we know we have new regions. And then another thing about the human brain is that there's a lot of variability in the cortex. The size of regions is different in different people, maybe by a factor of 2. And and if you look at a brain and it's all its folding and structures and the way the gyruses and the sulci form, those are different between different people. You can even tell identical twins apart by the way their brains are folded, which argues that it's not all genetic either.
Yeah, well, that, and also, I guess the significance of you're talking about size. And so maybe listeners will say, well, okay, cool. Like our brains are bigger. Great. Or we have a few more parts. Cool. I guess the thing about it is, well, how much of that, like what are those parts doing and those connections are doing? So this is where we get to the specifics of inhibition, abstraction, intelligence, certain types of emotional responses, et cetera. Is it these, the sizes are relevant based on what the functionality can do or not do based on other primates, or how do we understand that? Like, because obviously, like for example, chimpanzees can do some form of executive functioning, right? Sequencing, planning, organizing, but we can do it at a very high level, or we can do 3 times or 4 times more. So it's not like they can't do it. It's the fact that we can do much more or at a much higher level. All of that.
Well, that's a pretty deep question. And I can only give you a very sketchy kind of thoughts on it. But if you look at the way the human brain is different than a chimpanzee brain is, for example, our primary visual area or our primary motor area hasn't changed that much. It's about the same size. In fact, more of a chimpanzee brain is about vision than ours is. But there are new areas, new regions in between these primary areas, which we call association areas. And those are the ones that seem to have grown particularly larger in human evolution. And what those regions do exactly isn't well known. But some are involved in executive functions in the front of the brain. Some are involved in extrapersonal space. Some are involved in understanding language and that kind of thing.
Yeah, it's interesting because you think of the functions that we know. Again, sometimes there's a little bit. I think there's a little bit too much emphasis placed on localization in the brain, right? And what we know about the brain is that it's a web, it's a network, and everything's connecting and it's humming at the same time. But there are certain elements of social cognition that have to do with the orbital frontal sections of the brain, or certain things that have to do with anterior cingulate. Sure. Those functions have difference for us as humans that they do not have in chimpanzees or rhesus monkeys or things like that.
I don't know that's true. I mean, you're assuming that's true. I don't know if that's true. People doing experiments in monkeys want to find out, do monkeys get depressed? Is it the same parts of the brain that make them depressed? How do we treat that in a monkey? What's going on? So it is really hard to, it's hard for me to just agree with what you just said. Exactly. But it's fascinating.
Yeah, I guess my thing is that sometimes it's not always clear of, well, it takes, so let's use an example like language, right? This is something that people make a big to-do about, right? Is that we speak and we can, have, words and symbols and sounds, etc. Sure. But all animals, well, not all, I don't want to say that, but there are a handful of animals that will communicate in certain ways. They're, especially, for certain mammals. I mean, obviously chimpanzees communicate, dolphins and orcas, and, there's some form of communication. Something is going on. So because they don't use language in the way that we do, does not, make us necessarily, special or better. But what is it about language, I guess, specifically as a kind of example here that kind of sets it is a huge, as far as we know, in the same way in which we do, there's no other animal that has, alphabet and language, and as far as we know, how do we make sense of how that sets us apart from other animals?
Yeah, it's a good question. We do know that it's hard to teach an ape to speak human, even with sign language. They can do a certain amount. We do know that children who are deaf can learn to sign language beautifully and have beautiful language. We do know that in the human, there's regions of the brain that are involved in language. And the first one was found by a guy named Broca, who was a French neurosurgeon, who discovered a patient named Tan. And Tan could only say one word, and that was his own name. He could understand everything, and you could ask him to subtract 9 from 13, and he would say tan, 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 tan to indicate 4. So he could understand things, but he couldn't say stuff. And when this patient died, Broca looked into his brain, and there was a region of the brain, which is now known as Broca's area, which was damaged in this patient. And since then, lots of patients with damage in that area have shown defects in the ability to speak language. And since then, there's been a lot of research on the brain areas that are involved in language. And it's known that even when a baby is born, some of these areas are already developed. And in humans, for example, Broca's area is big on the left side of the brain generally. Whereas in a primate, like a chimpanzee, it's about the same size on the left and right, and it's not particularly big. But in a human, it's particularly big, 7 times bigger than in a primate. What that area is doing, how it works, we don't know, but we know it's involved in, generating speech. And there are other areas that are involved in understanding speech and understanding written words. And we also know that those seem to be connected up in preferential ways, even when a baby is born. So it's ready to learn language. Of course, a baby isn't born able to speak complex sentences with complex grammar. It learns how to do that, and it needs to learn it during a particular early period of its life, where it probably won't get the circuitry refined enough so that it's able to speak well.
Yeah, you're referencing I believe, Broca's and Wernicke's areas. One is for expression of language, one is for receptive language, and what you were mentioning earlier was the aphasias that people have, of how there are some deficits. And I think that for a long time there was, again, kind of what I said earlier, there's this kind of fascination of this localization. It's like, language is in these 2 areas, but language is in different parts of the brain. It might be primarily,
many areas.
Which is, but there is a distinction.
Sometimes language is on the right side, and sometimes it's on the left side. Sometimes it's on both sides more equally. And one of these things about asymmetry, the left-right thing, is that when a function is strong on one side, it's usually weaker on the other side. And Sperry, the same guy who brought us chemospecificity, studied split-brain patients and found if they were good at something on the right side, they were probably less good on the left side, and vice versa.
Well, I guess the last question I have is there's many things about the brain, and so you've written a wonderful book, I think, for that can be really, really helpful for a lot of people. And I guess my last question is, what is it that you want people to take away from the book? If there's anything to say, yes, you got it. That's exactly what I was hoping. You as the reader, that's what I wanted you to get from the book, and you definitely got what I was trying to say. What would that 1 or 2 or things be that you would want people to get out of it?
I guess I would want people to understand that by the time a baby is born, a lot has happened in the making of a brain, although you don't see it. Many people think perhaps all brains are the same and everybody is, absolutely equivalent. This kind of goes back to Steven Pinker's book called The Blank Slate, The Denial of Human Nature. in which there's a feeling, and I think this was brought home to me, and that was one of the reasons I was interested in writing this book, his book, that was really stimulating, and also some questions that I had from members of my family, saying, now if I just work at this, I can be a pro tennis player. If I just work at this, I can do this. And not acknowledging that everybody has a different brain from everybody else. Mm-hmm, mm-hmm. And that's already happened by the time you were born. It's not just your genes that have done it, but it's the experience your mother's had and the traumas or not that she's been through. It's due to the experiences you had as a child when your neurons were very plastic and changing. So everybody has this brain that's made, and it's, even at the time of birth, it's a different brain than everybody else's.
Yeah.
So everybody's an individual in that sense.
Yeah, that definitely comes through, especially there's a kind of like a point at which, kind of at the end of the book, it kind of ends to this point of like, yes, here's all of these things kind of through the neuron's eyes. And then here you go. Here's how we understand some things about us as humans, which I think is super important and there's a lot of potential implications in positive ways for people. Well, the book is called Zero to Birth, How the Human Brain is Built. It's fantastic. I hope everybody picks it up and reads it. Bill, where can everybody find you? Where can they get the book? Where can they find your research and find you online? All that good stuff.
Princeton University Press is the press that publishes the book. You can find it on Amazon. I work at Cambridge University, where I still teach. And I'm a developmental neurobiologist. I study mostly fish and the way that fish brains develop, but I use that as a model for understanding basic principles of brain development.
Yeah. Well, look, I mean, this was too much fun. I had so much fun talking to you about this stuff, about the human brain and getting all of your wonderful knowledge just downloaded here. So I really appreciate it.
I appreciate all your questions. You are like a student, like I supervise students and they come up with questions like you do. So it's great. But I'm glad that you were able to appreciate the book. Thank you.
Absolutely. Thank you.